Hello, my name is Professor Hudson. Let us continue with section 12.2, which deals with the structure of DNA. As mentioned prior, the DNA molecule consists of two individual strands of nucleotides that are hydrogen bonded together via the complementary base pairing. The DNA model consists of the two strands assembled like a twisted ladder, with the bases protruding inward towards each other from the sugar phosphate backbone like rungs on a ladder. Hydrogen bonds hold these base pairs together. The two strands in DNA double helix are said to be antiparallel. That is, they are oriented in opposite directions. From one end of the DNA strand molecule, if one strand starts with the free sugar and ends with the free phosphate, the other strand starts with the free phosphate and ends with the free sugar. Okay. Let's watch an animation that illustrates DNA structure.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar-phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
Okay, let's review. Question 3. Compare DNA structure with a ladder. The sides are the and the rungs are the. Choose the correct answer. Question 4. When DNA ladder's rungs are the same width, why do A and T and G and C form complementary base pairs according to Chargaff's rule? Choose the correct answer. Question 5. If a segment of DNA contains 430 nucleotides and 120 of the nucleotides are guanine, there will be how many of its complement cytosine? Now that we've talked a little bit about the structure of DNA, let's take a look at how exactly does DNA encode genetic information. Genetic information is encoded in the sequence of nucleotides. The key lies in the sequence, not the number of subunits. Within a DNA strand, the 4 types of bases can be arranged in any linear order, and this sequence is what encodes genetic information. The genetic code is analogous to language, where small sets of letters combine in various ways to make up many different words. English has 26 letters. Hawaiian has 12 letters. The binary language of computers uses only 2 letters. In the case of DNA, the language of genetics is dictated by 4 letters: A, T, G, and C. Now let's take a look at DNA replication and how it ensures genetic constancy during cell division. DNA replication produces 2 DNA double helices, each with an original strand and 1 new strand. When replication is complete, each parental strand and the daughter strand that was copied from it by base pairing wind together into a new DNA double helix, thus creating 2 copies of the original double helix. Base pairing is the foundation of DNA replication. An A on one strand will pair with a T on the other strand. A C will pair with G. If a parental strand, for example, read TAG, then the complementary new strand should read ATC. Let's watch an animation of this.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the 2 complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, 2 different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer.
As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
So let's continue talking about DNA replication. In the 1850s, Rudolf Virchow, an Austrian pathologist, realized that all cells come from pre-existing cells. Cells reproduce by dividing in half. Most body cells do this through by mitosis, and then sex cells divide to make gametes in meiosis. Each of the daughter cells must get an exact copy of the parent cell's genetic information if it's mitosis with body cells. Therefore, duplication of the parent cell DNA must occur before cell division. Duplication of the parent cell DNA is called DNA replication. DNA replication produces two DNA double helices, each with an original strand and one new strand. The fact of complementary base pairing suggests a model for how DNA replicates. The ingredients for DNA replication are threefold: 3 parental DNA strands, biparental DNA strands, free nucleotides, a variety of enzymes that unwind the parental DNA double helix, and synthesis of the new DNA strands. Let's take a look at how this occurs. This video is going to show you a view of DNA replication.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.